Hello and welcome back. Today we're going to continue talking about evolution, and in particular we're going to discuss the formation of new species. So by the end of this video you should be able to do the following: define a species and describe how scientists identify species as being different from each other, describe genetic variables that lead to speciation, identify prezygotic and postzygotic reproductive barriers, explain allopatric and sympatric speciation, describe adaptive radiation, describe pathways of species evolution in hybrid zones, and explain the two major theories on rates of speciation. So first, what makes something a species? Okay, how do we define what is a species? For example, I'd just taken a glance at these organisms, this bird and this bird, look very similar to each other. Their structure is very similar, their appearance is very similar to each other, but they're actually two different species. In contrast, this organism here, which is a type of dog, and this organism here, another type of dog, are actually the same species. And in fact, they can mate with each other and make offspring. So we can't rely upon something's appearance to determine whether or not it's the same species. So how do we define a species? Well, the biological species concept, the way we define a species is a group of organisms that can interbreed in nature and produce viable, which means living, and fertile offspring. Fertile meaning that offspring can have kids of their own. Another definition that would be important is gene pool, which is a collection of all the variants of genes in a species. One way of diagramming speciation events or how different species evolved is by using something called a phylogenetic tree or an evolutionary tree. Okay, this is an example of one that Darwin drew out in his book on the origin of species by means of natural selection. And this is one that's diagramming elephants, modern elephants, the Asian elephant and the African elephant, and their ancestors. Now, we're not going to learn details about how to read the phylogenetic tree right now. We are going to learn that in later lectures. So, what is speciation? Well, that's the formation of two species from one original species. In order for speciation to happen, there's some steps that need to occur. First, two new populations must form from one population. The two populations are reproductively isolated, so basically they stop mating with each other, so they start to form two separate gene pools. Each population will then adapt or evolve, and they're going to adapt or evolve differently from the other population. And then because of that, individuals of the two populations are no longer able to interbreed with each other. Now, for speciation to happen, there's two different ways it can happen, which is called allopatric speciation or sympatric speciation. Okay, allopatric speciation is when the gene flow between what's going to become two separate populations is interrupted by a physical barrier. Okay, so you have some original population of organisms. Then there's some geographic barrier isolating them. Okay, that could be, for example, let's say you had a pond and there was a drought and the water levels went down in the pond, and then that created two separate ponds where the fish are now separated from each other. Okay, so there's a geographic barrier which then produces reproductive isolation. The fish over here can no longer mate with the fish over here. Okay, over time these two populations start to diverge differently from each other, and you get two separate species evolving. What this geographic barrier is going to depend upon the organism. For example, the Grand Canyon would definitely be a geographic barrier for like a squirrel. Okay, a squirrel on one side of the Grand Canyon can't get to and therefore mate with a squirrel on the other side of the Grand Canyon. But that same barrier, the Grand Canyon, would not be a geographic barrier for a hawk. Okay, the hawk can easily fly from one side to the other. Now, the further the distance between these organisms due to this geographic barrier, the more likely the speciation is going to occur, because it makes it less likely for the organisms to be able to mate with each other. Multiple isolated habitats result in adaptive radiation, which I'll describe that one in a minute. So first, allopatric speciation. Okay, here's an example. Okay, the northern spotted owl, okay, and the Mexican spotted owl. they inhabit different geographic regions. Okay, northern spotted owl lives in these areas. The Mexican spotted owl lives in these areas. Okay, so they're separated by this distance between them. Because of that, those owls are not regularly mating with each other and exchanging their gene pools. These owls are an example of incipient speciation, which means they're in the process of diverging into two separate species. Speciation can occur when groups become isolated geographically for a long period of time. Changes, which are mutations in the DNA, in each group over time lead to the groups no longer being reproductively compatible, so they stop having the ability to mate with each other. Dispersal is when a few members of the species move to a new geographic area, whereas vicariance is when a natural situation arises to physically divide the organisms. Okay, so for example, one lake being separated in two lakes by a land mass between them, or two land masses separating away from each other. If you're talking about it on a large geological time scale, like for example, Australia moving away from the other continents. Now, you can also get adaptive radiation. Okay, that's where one species, okay, diverges into many different species. Okay, why? Because these different species are under different selective pressures, so they start to diverge away from each other. One way is based upon their source of nutrition, their food source. Like this bird starts to focus on eating seeds, and so it develops a large, powerful beak to be able to crack open those seeds. Whereas this bird starts to be able to take nectar in from flowers as a food source, so it has to have a long beak to be able to get to that nectar. The other type of speciation you can have is called sympatric speciation. That's speciation within the same physical location and conditions. Okay, so there is not a geographic barrier isolating them. Gene flow interrupted without a physical barrier can be due to errors in cell division or chromosomal structure, habitat differentiation, so them inhabiting some new ecological niche, or sexual selection. I'll give you some examples of that. The barrier is often genetic incompatibility. So in other words, there is no separate geographic isolation. You have a population of organisms, and then within that population, you start to get a separate population diverging until they end up being two separate species. Allopatric and Sympatric sound kind of similar. If you're trying to remember them, one kind of pneumatic way of remembering it, think same. Okay, so sympatric speciation is organisms that are living in the same area. They're still around each other. They're not separated by a physical barrier. Okay, ways that you can get sympatric speciation. One is errors during cell division leading to aneuploidy. So in Bio 1, you learned about aneuploidy. That's one too many or one too few of a chromosome. Okay, and that can have severe consequences if you're talking about like a mammal, like a human. But some species that are more capable of handling an extra chromosome compared to humans. Okay, but that's one way of getting sympatric speciation, is this error during cell division that leads to a different number of chromosomes than the species originally had. Or another error during cell division is called aneuploidy. Okay, that's when you have an entire extra set of chromosomes. So for example, let's say you start with an organism that's diploid. Remember, that means they have two of every type of chromosome. Now, an error happens during cell division where the chromosomes don't separate. They actually stay together. And then you can get an offspring that is polyploid, in this case tetraploid. Instead of having two of every chromosome, it has four of every chromosome. Now, again, we can't do that as humans. That would be lethal for us. But plants do that just fine. Okay, a lot of plant species are polyploid, more than two of each type of chromosome. And when you change the chromosome numbers, that can lead to a new species evolving. Another error during cell division is allouploidy. Okay, so this is when two different species mate to produce viable offspring. And for example, one species has, let's say, 3 different types of chromosomes. It makes its gamete one of each type. The other species has 2 types of chromosomes. An error happens. So the gamete has 2 of each type of chromosome, whereas it should have 1 of each type of chromosome. These combine, and then it mates again with a normal gamete, and that will end up with this new species that will have 5 different types of chromosomes instead of the 3 that this species had or the 2 that this species had. Again, you're not going to see that happen in humans. Okay, that would be lethal for us. But again, plants can do this just fine. Okay, it can produce viable and fertile offspring. So an example of sympatric speciation. Okay, this one due to habitat differentiation, so some new ecological niche. So these cichlid fish, okay, in Nicaragua show evidence of sympatric speciation. There's this lake that was populated about 100 years ago by a single population, so species of this cichlid fish. However, nowadays there are two populations with distinct morphologies, so they have different body plans. The easiest way to see it is looking at their lips here, thin-lipped versus thick-lipped. They eat different diets, and scientists believe that this is in the early stages of speciation, so these are starting to separate into two separate species. Another example is sympatric speciation. Sympatric speciation can be due to sexual selection. So for example, in this lake, Lake Victoria, you have these species of fish, and what happened is that the females started selecting the males preferentially based on their colored pattern. Some females preferred the males that had this appearance, whereas some females preferred the males that had this appearance. Over time, they started to select only this one or only this one. So because of that, they didn't exchange their gene pool between these two different physical appearance of fish. Okay, because of that, these two fish are starting to diverge away from each other into two different species because now their gene pools have been separated due to sexual selection. The females are picking which males they want to mate with. All right, so how do we get reproductive isolation? Well, there's two types of barriers, prezygotic barriers and postzygotic barriers. Okay, so first we're going to talk about prezygotic barriers. Okay, prezygotic means before the zygote forms. The zygote is when the sperm and the egg fuse together. Okay, so prezygotic blocks the fertilization, can occur by impeding different species from attempting to mate with each other, so they don't even try to mate with each other. By preventing the successful completion of mating, so they try to mate, but it doesn't quite work. Or hindering fertilization if mating is successful. So they do mate with each other, but the sperm cannot fertilize the egg. Okay, then there are different types of prezygotic isolation. This would be temporal isolation, habitat isolation, behavioral isolation, mechanical isolation, and gametic isolation. We're going to run through each of those with an example. So, types of prezygotic isolation. First one, temporal isolation. So these two different species of frogs, they live in the same area. But the different species of frogs, because they don't mate with each other. Why don't they mate with each other? Because they mate at different times of the year. One species only mates around February, the other species only mates around April. So because of that, they won't ever mate with each other, therefore they won't exchange genetic information, there'll be two separate gene pools, and because of that they diverge into two separate species. So temporal, time, okay, so different times of year, or it could be different times of day, some mate only during the day, some mate only during night, etc. Okay, that's one type of prezygotic isolation. Next type of prezygotic isolation is habitat isolation. Okay, so these two different species of crickets, they live relatively close to each other, but one species prefers sandy soil, the other species prefers loamy soil. So because of that, they're living in different habitats. So because of that, this one doesn't go over to this area, so it can't mate with this one. This one doesn't go over to this area, so it can't mate with this one. So again, they don't attempt to mate with each other. Why? Because they live in different habitats. That's habitat isolation, which is a type of prezygotic isolation. The next type of prezygotic isolation is behavioral isolation. So some species will only mate with another member if they perform some mating ritual. So for example, these blue-footed boobies, they do a little dance, okay? And that dance is what convinces the other member of its species to mate with it. If it doesn't do that dance, then it won't mate. So if they don't have that particular behavior, then they won't mate with each other. They choose not to mate with each other. So that is an example of prezygotic isolation. They don't even attempt to mate with each other. Why? Because they don't do the right dance. Another type of prezygotic isolation is mechanical isolation. These are morphological differences or structural differences that prevent successful mating. So, for example, these two snails here, they're mating with each other. Okay, these two snails are attempting to mate with each other, but they can't. Why? Basically because their gonads, where you find their gonads, they can't line up with each other correctly. Why? Because of the shape of their shell. Okay, you can see, this shell are both clockwise. This shell is counterclockwise. This shell is clockwise. So because of that, these snails physically cannot line up in the correct orientation to be able to mate with each other. So they mechanically just can't do it. They can't mate with each other. So that's a type of prezygotic isolation. Last type of prezygotic isolation is gametic isolation. That is where the gametes cannot fuse together to form a zygote. Okay, so for example, you have these sea urchins, these purple sea urchins, these red sea urchins. Now the way sea urchins mate is they just release their eggs and their sperm into the water around them. So if that happens, these sea urchins are right next to each other. So they're releasing the eggs and the sperm into the ocean around them, and those eggs and sperm are near each other, but the sperm of one species is not able to fertilize the eggs of another species. Okay, they cannot complete fertilization. So even though the gametes are with each other, it just physically won't work. So that's gametic isolation. So those are all examples of prezygotic isolation. So things that separate different organisms from exchanging genetic information in order to keep them separate species before the zygote forms. The other type of reproductive isolation is postzygotic. So that is after the zygote forms. So when the zygote is able to form, the sperm can fertilize the egg, but then something happens. Okay, things that can happen is hybrid inviability, hybrid sterility, or hybrid breakdown. So again, some examples of those. Hybrid inviability. That's where two species mate with each other and they produce an offspring. That offspring is inviable. In other words, it won't live, it dies. Okay. Since it's dying off, you're not really passing on your genetic information to the next generation. So this keeps them as two separate species. Next example is hybrid sterility. So if you, for example, take a horse and mate it with a donkey, they can and will mate with each other, and that produces a mule. And the mule is viable, it's living, but it's infertile, it's sterile. The mule cannot have kids of its own. So because of that, the horse and the donkey are two separate species. Remember, the biological species concept says that two organisms have to be able to mate, produce viable, which is living, which this does, and fertile, okay, offspring. Well, the mule is not fertile, so they are two separate species. Last example of postzygotic isolation is hybrid breakdown. So that's where two separate species are able to mate with each other, produce a viable offspring that is fertile. Okay, however, these offspring, when they mate with each other in the next generation, it breaks back down. It produces an inviable offspring. Okay, so basically it takes an extra generation for it to break down, so that's why it's called hybrid breakdown. But again, they can't produce viable and fertile offspring, at least after a couple of generations. So hybrids are when two separate species are able to mate with each other. For example, a horse and a donkey making a mule. Or for any Napoleon Dynamite fans out there, a liger, which is a lion and a tiger mating with each other to produce a liger, okay, which is a hybrid, a combination of two different species. Again, it can be infertile, okay, that's why they remain two separate species. So that's what hybrids are, the progeny of two different species able to mate with each other. So what happens when you get hybrids? Well, there's a few things that could happen. So one thing that could happen is reinforcement. So you get two separate species, okay, so one species is going this way through time, the other species is going this way through time, but they are able to mate with each other and produce hybrids, okay, but the hybrids are less viable or less fertile than either of the two species. So what happens is these species keep diverging farther away from each other, and the hybrids start to die off, okay. So that's reinforcement. You reinforce the separation of the two species. Another thing that can happen is fusion. So you start to separate two separate species, but they're able to produce hybrids. And it turns out these hybrids are also just as healthy as the two separate species. So what happens is the two species and the hybrids are all exchanging genetic information, so they combine back into one gene pool. So they fuse back together. The third thing that can happen is stability. So you start to separate into two separate species. Now those two species can mate with each other and produce hybrids. But let's say that these two separate species, they're living in two geographic areas, and there happens to be a region where those two geographic areas meet. Okay, in that case, they can keep producing those hybrids when they happen by chance to meet with each other. But since the hybrids are not as viable as two separate species, it won't fuse back together. Okay, so it will remain like that. You have two different populations. Occasionally they meet and can produce hybrids. When you're talking about species, something to note is how fast does speciation occur? Okay, well, there's two different ways that you can think about this. One, the way that Darwin thought about it, is gradual speciation. Okay, so species diverge gradually through time through many small steps. Okay, kind of look at it in a graph form like this. So as time passes, okay, you get more and more evolutionary change in the species. So it's kind of a steady rate of how fast that species diverges over time. Okay, but we've seen that we don't always have this gradual speciation. We can also have punctuated equilibrium. That's where species exhibit a large change in a relatively short period of time, followed by long periods where it doesn't change. Okay, so that's, for example, here, time is passing, there's not much change, and then you get a change. And then not much happens, then you get a change again, a big change, and then it passes through time without much change, and then another change. Okay, or another way to visualize this. Okay, gradual speciation, you have one founder species, it starts to diverge into two separate species, and over time you get slow changes, slow gradual changes to the organisms to develop those two different species. One beak becoming longer and thinner, for example, let's say to feed off insects or nectar. One beak becoming thicker, for example, to be able to crush seeds, to be able to eat seeds. Okay, or you can have punctuated equilibria. That's where there's a relatively rapid change. Now remember, rapid means on an evolutionary time scale, so it doesn't necessarily mean overnight. It could still mean thousands of years. But a relatively rapid change and then little change over time. Okay, so that's gradual versus punctuated equilibria. Okay, so in this lecture, in really brief summary, what we basically did is we talked about what is the definition of a species. Remember, in our case, how we're going to define it is to be the same species, the organisms have to be able to, in nature, mate with each other and produce viable and fertile offspring. Now, of note, that's not the only way to define a species. That's called the biological species concept. It doesn't work for everything. For example, you can't use the biological species concept to define different species of fossils, of extinct organisms, because you can't see if those fossils are going to mate with each other and produce viable and fertile offspring. Okay, or for organisms that reproduce asexually, like bacteria, okay, you can't tell if they can mate with each other and produce viable and fertile offspring because they don't mate with each other. So, for this course, we're going to focus on the biological species concept. There are other ways of defining species, but we're not going to focus on that. Besides defining a species, the other things we talked about in this lecture is how do we get new species? Allopatric speciation, Sympatric speciation. What are the reproductive barriers to isolation, prezygotic, postzygotic? So that concludes your lecture on the formation of new species. Until next time, this has been Dr. Sage.